Okay, so welcome back to this next video in which we are discussing the epidermis. So we're in the process of looking at the structure of the desmosomes, and we're doing it in a little bit more detail. We're looking at the domain structures of the placoglobin protein, the desmoplakin protein, and the placophilin protein. Okay, so let's now look at the domain structure of the desmoplakin protein. Okay, and this goes for both of the two splice variants of desmoplakin. So here's the amino terminus. You then have a portion that's known as the head, okay, and I'm having to draw this quite small because I haven't got limited space here. Okay, so we'll have the head coloured in pink here, so this is the head domain. Then you've got a portion called the rod domain, okay, which will consist of alpha helices in a coiled coil domain. Okay, so here we have the rod domain in blue. Then after the rod domain, you have three domains that are known as plakin repeat domains. Okay, so one of them is known as the plakin repeat domain A, the second is known as the plakin repeat domain B, and the third is known as the plakin repeat domain C. Okay, so these are what are known as plakin repeat domains. Okay. And for short, plakin repeat domains are abbreviated to PRDs. Okay, so P for plakin, R for repeat, and then D for domain. Okay, so I'll colour in the plakin repeat domain A in orange, the plakin repeat domain B in orange, and the plakin repeat domain C in orange as well. Okay, so we have three plakin repeat domains, and then after the plakin repeat domain C, we then have a region that is known as the GSR region, which is the portion that's going to be extremely important for binding to the intermediate filaments, specifically keratin. Okay, then you have the C-terminal tail. Okay, so the GSR portion. Now, let's start by showing this thing binding to our placoglobin protein here, and then we'll discuss the GSR region in a little bit more detail, because that's the portion that's then going to interact with the intermediate filaments. Okay, right, so the head portion is going to bind to the central armadillo domain of the placoglobin protein. So here's the amino terminus. Then we've got our head portion binding to the central repeat domain. Then we've got the rod portion, followed by the three plakin repeat domains, which I'm just going to combine into one there. So there are the three plakin repeat domains. And here then, this is this GSR region. Okay, now I want to discuss what GSR stands for. Okay, so GSR is going to stand for glycine, serine, arginine rich domain. Okay, so in order to understand why that is abbreviated to GSR, you need to be able to read single letter amino acid code. Okay, so each of the amino acids is given a letter basically. Okay, and the letter for most amino acids is just the first letter of the name. So glycine, for instance, is G, serine is S. Okay, and then the reason arginine is not A but is R is because alanine was given A. Okay, so if you read arginine, what letter would be the best second choice? Well, it is R, because you can hear arginine there. Okay, so that's why they gave it the letter R. So this special domain here, this GSR domain, okay, stands for the glycine, serine, arginine rich domain. Okay, so it's going to be rich in glycine, serine, and arginine amino acids. Now, let me just discuss the structures of these amino acids. We've seen the structure of glycine earlier when we were discussing the structure of collagen. Okay, let's now see the structure of serine and arginine. Okay, so here's the amino group. Here's the alpha carbon with a hydrogen coming off it. Now, we'll start with serine here. Serine is going to have a methylene group as its R group, then with an alcohol group coming off that methylene group. Okay, and then it will have its carboxylic acid group down here. Okay, so that's the structure then of a serine amino acid. Arginine's a little bit more complicated. Okay, it has a very long R group. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, and now the R group of arginine consists of, firstly, 3 methylene groups. Now, because I don't want to have to draw 3 methylene groups out, what I'll do is draw 1 methylene group, bracket it like so, and then subscript it 3. That's a useful trick of getting out of having to draw 3 separate methylene groups. Then we have the nitrogen atom with a hydrogen coming off it, and then a carbon atom. Okay, this carbon atom then has an amino group coming off it, and a double bond to a nitrogen atom, which then has a hydrogen coming off it. Okay, so that's the structure of the amino acid arginine here. Okay, so this GSR region is rich in glycine residues, it's rich in serine residues, and it's rich in arginine residues, and it's this region which is believed to be the region which interacts with the intermediate filaments, such as keratin. Okay, right, then all we need to discuss now is the structure of the final protein involved here, which is the plakophilin proteins. Okay, so firstly, let me remind you that we discussed the fact that there were going to be 3 different genes for plakophilin proteins. Okay, so remember, plakophilin is often abbreviated to PKP for short. P for pla, K for co, and then another P for filin here. Okay, right. And there are 3 different genes for plakophilin proteins: plakophilin 1, plakophilin 2, and plakophilin 3. Okay, now for the plakophilin 1 and the plakophilin 2 genes, there are 2 different splice variants. Okay, in both cases there is an A splice variant and a B splice variant, which overall means that there are 5 different plakophilin proteins. There's plakophilin 1A, plakophilin 1B, plakophilin 2A, plakophilin 2B, and then plakophilin 3 is the only one that doesn't have 2 splice variants, so it's just plakophilin 3. Now, unfortunately, the naming here is the opposite way round to the way it is for the desmocolin splice variants. The A splice variant is the short splice variant of plakophilin, and the B splice variant is the long splice variant of plakophilin. Okay, right, but again, all the important domains are present in both splice variants, so for our drawings we're not going to see any difference between these different splice variants. So all plakophilin proteins are going to have the same basic structure. Okay, and I think I'll draw it actually on this picture. So the domain which is actually going to interact with the head domain of desmoplakin is also going to be the head domain of our plakophilin protein. Okay, so in light green here, this is the head domain of our plakophilin protein interacting with that head domain of our desmoplakin protein here, and it will then also interact with the head domain of another desmoplakin protein here. Okay, so remember that plakophilin's role is to link 2 desmoplakin proteins together. Okay, and it's going to do this via its head domain, which will interact with 2 head domains of desmoplakin proteins. Okay, right. Then, plakophilin is actually also an armadillo family protein, so it's going to contain armadillo repeat domains. It doesn't quite have as many as plakoglobin, it only has 9 of the things. Okay, so firstly it's going to have 5 of the things. Okay, all in tandem. Then it will have a little spacer, and then it will have 4 more of these armadillo repeat domains here. Okay, after that it will then have the tail domain here, and then it will have its C-terminal tail, its C-terminus facing up towards the cell membrane, like so. Okay, so these here, these are all armadillo repeat domains, so we've got 9 in total. Okay, and then right over there we've then got the tail of our plakophilin protein here. Okay, so let's just label this up. So this blue portion here, this is the tail of our plakophilin protein. This green portion here, which is interacting with the heads of the desmoplakin proteins, that's the head of plakophilin. Okay, right, so that's a little bit more of an in-detailed study then of the proteins involved in attaching these desmosomal cadherins to the intermediate filaments of the cytoskeleton. Now, let's go back to the bigger picture. So, we have these huge aggregations of these interactions between desmosomal cadherins. okay? And it's often compared to Velcro. Each interaction between two desmosomal cadherins is actually extremely weak. So to get a strong interaction, you have to have a huge number of them aggregated together, which are interacting with a huge number of them aggregated together on the other cell, okay? And this huge cluster of desmosomal cadherins interacting with desmosomal cadherins on the other cell, this is known as a desmosome. So the entire thing here, this is a desmosome. Okay, right. And it's these things which you can see as little spines on these prickle cells of the stratum spinosum. Okay, right. So that's the characteristic feature that's very, very obvious in the stratum spinosum of the skin. Let's now work our way up to the next layer of the skin. Okay, so we're now continuing on the differentiation process, and I want to stress that again, that as you go up the different layers of the epidermis, the cells are gradually going through a differentiation process. Let's draw this out again. So here is a rete ridge, and here's a rete peg. Okay, remember that the bottommost layer is the basement membrane here. Okay. Then sitting atop the basement membrane, we have the basal layer of cells. Okay, and remember this consists of two different cell types. It consists of the epidermal stem cells themselves, which remember are these cells which are not yet differentiated. Okay, these are not committed to differentiate. However, they are unipotent stem cells, so they can only become one type of cell. Okay, so here are the epidermal stem cells. Then what will happen is an epidermal stem cell will divide into two daughter cells. Okay, and each of these daughter cells gets the option of whether it wants to remain a stem cell, okay, or whether it wants to now undergo the differentiation process. So, roughly half of the daughter cells remain epidermal stem cells, and that means that you maintain your population of epidermal stem cells over time, and then the other half will then go on through the terminal differentiation process. Now, differentiation is a slow process, okay, and all the way up the epidermis, up the multiple layers of the epidermis, you are getting closer and closer to the final goal of being a differentiated cell, okay? So differentiation goes on all the way up whilst you're moving up, okay? So, the first stage that you go into after you've just decided that you're going to differentiate is to become one of these transit amplifying cells, which is still attached to the basement membrane and which is still capable of dividing. You then divide and produce a whole population of other transit amplifying cells. Okay? These cells are also all committed to undergo the differentiation process. After a certain amount of time, you will cleave away from the basement membrane, and when you do so, you also lose your ability to divide, so the upper layers of the epidermis are not capable of dividing. Only the cells in this basal layer, the stratum basale, are capable of dividing. Okay, so you then move up into the next layer, which contains multiple layers of cells, okay, but it's just known as one layer. Okay, and these cells are not going to be attached to the basement membrane anymore. Okay, they are sitting atop the basal layer of cells, which are attached to the basement membrane. And these next layers, these are known as the stratum spinosum, which is the layer that we've just been discussing, the spiny layer of cells. Okay, and the characteristic feature of these cells is that they have a huge number of desmosomes, which are very, very visible, so they look spiny basically. All of the cells are attached to one another by these desmosomes. Okay, and this makes the layer look spiny, and you can see this down the light microscope. Okay, so you don't need an electron microscope to see this. Right, so that's the stratum spinosum. Okay, above the stratum spinosum, you then have the next layer, which is what we're going to now move on to. Now, this is a monolayer of cells, okay, so it's a single layer of cells that sits above this. and this is going to be called the stratum granulosum. Okay, and it's called the stratum granulosum because the cells in this layer appear very dark when you stain them. Okay, so I'm going to colour the stratum granulosum in vivid purple here, which is quite a dark purple. Okay, so here are the stratum granulosum cells, and this is the layer that we're now going to talk about. Now, the key important role of the stratum granulosum is that it's going to be responsible for waterproofing the skin. Okay, and the way it's going to achieve this is through, again, cell adhesion molecules. And we're finally going to discuss tight junctions in a moment, because these are the types of cell adhesion molecules that you're going to need in order to make this layer waterproof. Okay, so gradually what will happen is the cells of the spiny layer will undergo further differentiation, and they'll move up into this stratum granulosum here. And when they do that, they'll develop tight junctions that allow them to make this layer waterproof. Okay, so this is the next portion of this differentiation process of these keratinocytes. Okay, so let's now discuss tight junctions, because these are these things which are going to be responsible for waterproofing this layer. Okay, so basically the principle of a tight junction requires me to draw a three-dimensional picture. Okay, so let me draw a cell within this stratum granulosum layer here. So let's let this be a cell. Okay, so this is one of these cells removed here. This, let's say, is the side facing the external world. It's going to have another layer on top of it, but it's the side closer to the external world, and this is the side facing down towards the basement membrane. Okay, then let's have another cell here. Okay, so here's another one drawn in an attempt to show a 3D diagram, and then maybe we'll put another one back here as well. Okay, so here is a third cell right back there. Okay, right, so basically what we are going to do is if we want to waterproof this layer, we need to seal all of these gaps between the cells, because we don't want water to be able to get either into the body, and more importantly actually, we don't want it to be able to get out of the body, because otherwise water would be continuously evaporating from the body and you would hugely dehydrate. Okay, so you need to be able to seal these gaps between the cells, and this is what tight junctions are going to do. You are going to form basically an obstruction, a physical obstruction, all the way. You're going to fill in this gap by cell adhesion molecules. You're going to be putting cell adhesion molecules in between all of the cells here, and you're just going to fill in this gap. Okay, and this is the concept of a tight junction. Okay, you put loads and loads of cell adhesion molecules. You'll have one cell adhesion molecule on this cell, one on the other cell, and they will overall form a little barrier there. You'll do the same a little bit more along. You'll do the same a little bit more along. So you'll overall form this barrier that obstructs this gap that we would have in between the cells otherwise. Okay, and this is the concept of tight junction. All of these little cell adhesion molecule interactions which are overall filling in the gaps between cells. This is the concept of a tight junction. This is responsible for waterproofing the stratum granulosum. Okay, right. So tight junctions are often compared to the plastic packaging that you have in a 6-pack of beer or something along those lines. So if you go to the supermarket, you can buy 6 packs of beer which are packaged in a piece of plastic. Now, basically the cans will be loose, so they'll be like so, but they'll all be held together by this very, very tight piece of plastic, like so. Okay, so these are the beer cans, and they've all got their heads wrapped around this piece of plastic, this sheet of plastic. So you'll have 3 more cans there. Okay, and the piece of plastic fills in all the gaps between the cans of beer. So if you've got your finger here, you cannot get your finger to the other side by squeezing it in between the beer can and the plastic because it's so tight. That is analogous to what the tight junctions are like. Okay, the cells are analogous to the beer cans. Okay, and then the tight junction is analogous to the piece of plastic that wraps around the beer cans. Okay, so we now want to discuss the different types of cell adhesion molecule that are involved in the formation of these barriers that are tight junctions. Okay, so there are 3 different types of cell adhesion molecule that are hugely involved in the formation of tight junctions. Okay, now we're going to start off with most likely the most important, which are the claudins. Okay, so long ago when we were discussing the different families of cell adhesion molecules, I said that the 4 main families of cell adhesion molecules were the cadherins, the integrins, the selectins, and the immunoglobulin superfamily cell adhesion molecules. And that's true. Another family of cell adhesion molecules that's a little bit more minor is the claudins, but they're very important in the formation of these tight junctions between cells. Okay, now you have tight junctions in many other places in the body, not just the skin. Okay, now that's an important comment to bear in mind when I now tell you about how many different claudin proteins we have found in humans. Okay, so basically there are 27 known claudin proteins in humans. Okay, sorry, let me correct that. There are 27 genes for claudin proteins found in humans. Okay, some of these genes have multiple splice variants. Okay, it's rare for them to have multiple splice variants, but some of them do. Okay, I believe claudin 10 and claudin 18 have multiple splice variants. Okay, now the naming of them is very nice. Okay, the naming literally is just numbering. Okay, so usually you abbreviate claudin down to CLDN. Okay, and then it's claudin 1, claudin 2, claudin 3, all the way on to claudin 27. And as I say, claudin 10 and claudin 18 have multiple splice variants. I think each of them has 2 different splice variants. Okay, right. I think the 2 splice variants of claudin 10 are called claudin 10A and claudin 10B, whilst the 2 splice variants of claudin 18 are called claudin 18-1 and claudin 18-2. Okay, right. So those are all the different types of claudin. Now, they're not all found in the skin. The main one that's going to be found within the skin is, in fact, claudin 1, which is quite helpful. So claudin 1 is hugely found within the stratum granulosum of the skin. Okay, so let me show you the structure of these claudin proteins then. Okay, so all claudin proteins have the same basic structure. Okay, they all have their amino terminus on the cytoplasmic side of the cell membrane. So once again, these 2 parallel lines here represent the phospholipid bilayer of the cell membrane. Okay, now the polypeptide is going to span the membrane once to get onto the extracellular fluid side up here. Okay, then it's going to come back in, it'll span the membrane again, then it'll go back out, and then it will come back in. Okay, so they have these 4 membrane-spanning alpha helices ending with the carboxylic acid terminus intracellularly. Okay, right. So let's name up the portions of claudin proteins. So firstly, we have these 4 membrane-spanning alpha helices. Okay, now the 4 membrane-spanning alpha helices are known as TM1, standing for transmembrane 1. Okay. Then the second one is known as TM2, standing for transmembrane 2. The third one is known as TM3, and the fourth one is known as TM4. Okay, so that's the naming system of the 4 membrane-spanning alpha helices. Okay, then Other very important domains are these extracellular loops that we have here, because these are going to be involved in binding to another claudin protein that's on the surface of the other cell. Okay, so these are known as the extracellular loops. Okay, now for short, the extracellular loops are often abbreviated to the ECLs. So EC stands for extracellular. Okay, the E is for extra, the C is for cellular, and then the L is for loop. So we have two extracellular loops. We also have an intracellular loop here in orange. Okay, so that's known as the ICL for intracellular loop. Okay, in addition to the two extracellular loops and then this intracellular loop, other important domains include the amino terminal domain here, which is prior to TM1. Okay, so this is called the amino terminal domain, or ATD. So this stands for amino, because it's close to the amino terminus. Terminal, that's the T, and then the D is for domain. And then finally, we also have the C-terminal domain, which is this portion after TM4, which is close to the C-terminus. Okay, so this is called the C-terminal domain, or CTD for short. So C is for C, T is for terminal, and then D is for domain. Okay, right, so that's the structure then of a claudin protein, and all 27 of them have the same basic structure. So, now let's discuss the adhesion of two claudin proteins together. Okay, so if we have the cell membrane of cell 1 here, and we have the cell membrane of cell 2 here, it is the extracellular loops that are going to be important in the interaction between two claudin proteins. Okay, so here is one of our claudin proteins on cell 1. And now, whoops, I've drawn this one a little bit small, so the other one's going to have to be massive, basically, to actually be able to join to it. Okay, I apologise for that, that's a little bit out of proportion. They should have been equal sizes, ideally. Okay, so the key message to take from this drawing is that it's the extracellular loops which are involved in binding the two claudins together. Okay, right. So, the same question as always. Whenever you discuss cell adhesion molecules, you can't just talk about how they adhere together. You then need to discuss how do they attach to the cytoskeleton, because this isn't a viable, strong link between two cells. Okay, you need to attach a rigid structure here to rigid structure here. So these need to attach to cytoskeletal components, basically. So we've got these two claudin proteins attached together. If we're talking about the stratum granulosum, they're both mainly going to be claudin 1 proteins. Okay, how do they now attach to the cytoskeleton? Well, basically they attach to the cytoskeleton using proteins known as zonular occludins proteins. Okay, which are often abbreviated to ZO proteins. Now there are 3 different types of zonular occludins proteins. There is zonular occludins 1, there is zonular occludins 2, and there is zonular occludins 3. Now, these zonular occludins proteins, they bind to the C-terminal tail of the claudin proteins. Okay, so shown in vivid purple here, these are the zonular occludin proteins binding to the C-terminal tails of the claudins. Okay, so these are zonular occludins here. Right, and then the zonular occludins proteins are then going to bind to actin filaments of the cytoskeleton. Okay, so I'll just draw a little portion of actin here. Okay, so there's our actin filament, or the beginning of our actin filament, which, remember, consists of these 2 intertwined alpha helices of actin filaments attached to one another. Okay, right. So that is therefore how you attach claudin proteins to the actin cytoskeleton of the cell. Okay, now we'll call it there for this video, and in the next video what we'll discuss is other cell adhesion molecules which are involved in tight junctions.